Okay, so good evening, everyone. So I welcome you all to the week 11 more problem solving session of this NPTEL course Molecular Biology. So I will wait for a few minutes then I'll start today's session. So now we are almost approaching the end of this course. So I hope all of you have gone through the week 11 lectures. So the week 11 lectures are mainly dealing with genome editing and homologous recombination, non-homologous recombination. So, so there is quite a bit of information in week 11. So I'll try to break it up. So I will be taking another extra session on 14th of April. So on 14th of April, I will be covering the genome editing part. So today what we will see is we will mainly talk about the homologous recombination, non-homologous recombination, and a little bit part of genome editing. The rest part of genome editing, like CRISPR-Cas9 and talents and all these and the other tools for genome editing, I will cover it in the extra class on 14th of April. Okay. Also, in today's session, we will not talk much about the homologous recombination because that part was already covered. And if there is something remaining, I will cover it up in the extra class. So the molecular machinery of how homologous recombination takes place in prokaryotes and eukaryotes was covered in the video lectures. So I will not go through that part in much detail. Um, I'll wait for another 1 or 2 minutes and then start today's session. Okay, so I'll start today's session now. So let's share today's presentation. Okay, so I hope this screen is visible and I'm audible. Hello everyone. I'm Akash Jyoti Pathak, and I'm one of the PMRF TAs for this NPTEL course Molecular Biology. This course is being run by Professor Vishal Trivedi of IIT Guwahati. And today we are in the week 11 problem solving session or the tutorial session of this course, Molecular Biology. So first, let's have a quick recap of what all was covered in week 11. So week 11. In week 11, Uh, uh, the concepts that were covered were first of all we learned about homologous recombination. We started the discussion that was started with homologous recombination, how the molecular machinery of homologous recombination takes place in prokaryotes and eukaryotes. A part of this was also covered in earlier lectures. Uh, and thereafter we moved on to understand what is non-homologous recombination. And here in non-homologous recombination, there are several methods by which non-homologous recombination takes place. So we these were all covered one by one. So non-homologous enjoining, then conservative site-specific recombination, transposition. So here we dealt with the different transposable elements that are present in prokaryotes and eukaryotes, and then VDJ recombination. So if you have studied immunology, then you must have come across this VDJ recombination. So how this takes place in the immune cells to generate different antibodies. The variety of antibodies that are present in our body against the different antigens. How are these produced through this VDJ recombination that was covered, and then in the next lecture, the genome editing was discussed in quite detail. So starting from talons, then zinc finger nuclease and CRISPR-Cas9, all these were discussed. Okay, so what we will do, I will divide this week 11 lecture. So I will not cover everything or everything in this lecture, or some of the important concepts that were covered and are part of that might help you in solving your assignment. We will take it up in on 14th of April. So I'll take an extra class on 14th of April. So the genome editing part and a little bit about this homologous recombination machinery in prokaryotes and eukaryotes will be covered in that section. Okay. This homologous recombination is already covered. Okay. So let's start. First of all, we will see about the non-homologous recombination. Okay, so in non-homologous recombination, what is the meaning of non-homologous recombination? So when you are studying about meiosis, you must have read about homologous chromosomes. So what are homologous chromosomes? So homologous chromosomes are basically, so humans have 46 pairs of, or humans have 46 chromosome or 23 pairs of chromosome, right? So we all have 23 pairs of chromosomes. And how do we get these 23 pairs of chromosome? So one set of chromosome we get from the father, and another set of chromosome is obtained from the mother. Okay, so there are two copies of each chromosome. So if we talk about chromosome number one is coming from the paternal side and another is coming from the maternal side. So there are two copies of each chromosome. So these two copies of each chromosome are called the homologous chromosome. Okay. These are called the homologous chromosome. Why are they called homologous chromosomes? Because the structure of these two chromosomes will be very similar. The position of the centromere, the chromatid length, and the genes present in them, they will be very similar. However, there will be minor differences in the genes. So that is what is called as alleles, isn't it? So the minor variations in genes that are present, or the alternative forms of genes that are present. Okay, the alternative forms of genes that are present, those are called the alleles. Okay, so there are two copies of each chromosome, and during meiosis, what happens? You must have come across a term which is called as homologous recombination. Okay, so homologous recombination takes place during the prophase stage of meiosis one, or you must have come across this even in. So what happens? There is pairing of the homologous chromosome. Pairing of the homologous chromosome takes place at which stage of meiosis one? So in the prophase one. In prophase one, in the zygotine stage, there is pairing of homologous chromosome. There is formation of synaptonymal complex, and then in the Bacchitin stage, there is crossing over. So what happens is this is called what is exactly is homologous chromosome, homologous recombination. So in homologous recombination, the homologous chromosomes align with each other, and there is exchange of genetic material between the to homologous chromosome. Okay, which leads to so there is a stage called crossing over when this exchange of genetic material takes place, and this exchange leads to the generation of variation. Okay, so that is homologous recombination. Now here we are discussing about non-homologous recombination. So in non-homologous recombination, there is one concept which is non-homologous end joining. So what is non-homologous end joining? So whenever there is a double-stranded DNA break, to recover or to repair these double-stranded DNA breaks, there are several methods or several machinery where these double-stranded breaks are repaired. So one of them is non-homologous end joining. So here what is done is it does not involve extensive stretches of homologous sequences. So unlike homologous recombination, where the two homologous chromosomes will align, and these two homologous chromosomes has a lot of similar sequence of genes at the DNA sequences is almost is very similar in the homologous chromosomes. But here what happens is that particular aligning between these homologous regions does not take place. So there is no involvement of alignment of the extensive stretches of homologous sequences. Instead, what happens is that the two ends of the broken DNA are joined to each other by misalignment between single strands protruding from the broken ends. Okay, so we will see this in the next slide with the help of a diagram. So what happens is the ends of the broken. So first, the broken DNA that is produced that will have a free 3' end, and using this 3' end, these the at any region, if there is any homologous or complementary sequence, that will align and will and this region which is having this break will be aligned and repaired. Okay. So this misalignment occurs by pairing between tiny stretches of complementary bases. Okay, so what is happening in non-homologous end joining? There it will lead to error. Okay, it is error prone. Okay? Because randomly, what is happening? Randomly, alignment is taking place between any sequences. Okay, so if it finds any region which is complementary to that broken end, then it will use it as a template, and the new sequence will be generated. So these nucleases remove single strand tails and DNA polymerase fills in the gap. So there are a couple of proteins which are involved in this non-homologous end joining. So these are KU70, KU80, DNA PKCs, Artemis, XRCC4. So let's understand this through the figure. So you see here. So first you see this is the DNA. Okay. And in this DNA, a double-stranded break has been introduced. Okay, due to some mutagen or UV radiation or any physical agent, there has been a double-stranded break which is introduced in this double-stranded DNA. Now, now once there is this double-stranded break generated, of this protein is recruited, KU70/80. Now, this protein binds to this double-stranded break. So you see here a heterodimer of KU70 and KU80 binds to the broken DNA ends and recruits the protein kinase DNA PKCs. So these are protein kinases. So once this binds to the double-stranded DNA breaks, it recruits another protein kinase, which is DNA PKCs. And this DNA PKCs, what will do? What will it do? It will in turn recruit another protein, Artemis. Artemis is an enzyme. So it is an enzyme which is having exonuclease and endonuclease activity, which processes the broken ends. Okay. And then finally, a complex of ligase 4 with XRCC4 and XLF joins the broken end together. Okay. So this is the mechanism by which non-homologous end joining take place. Okay. So now answer this question. Artemis is a option A is polymerase, option B is kinase, option C is ligase, and option D is nuclease. So just now we discussed about Artemis. So Artemis is the enzyme which is recruited by the KU70 KU80 heterodimer. Okay. Okay, so what is happening in non-homologous end joining basically is the broken ends. These broken ends are ligated directly. They are ligated directly without the requirement of a homologous template. Okay, so the answer for this question just now we saw that it has nucleus property, Artemis. It has both exonuclease and endonuclease activity. Okay, so you see here, this Artemis is a has a 5' to 3' exonuclease and a latent endonuclease activity, and it is activated by the phosphorylation by DNA PKCs. So you saw here that first we were getting this, first this KU7080 heterodimer was recruited, and then this in turn was recruiting DNA protein kinases. This protein kinase was phosphorylating, was is phosphorylating Artemis, and once this Artemis is phosphorylated, it becomes activated. Okay, so the this so the so this is activated by phosphorylation. And who is doing this phosphorylation? This is done by DNA PKCs. And these nucleolytic activities process the broken ends and prepare them for ligation. Okay, for ligation you require a 5' phosphate end and a 3' OH group. Okay, so this Artemis is having this exonuclease and endonuclease activity, and with the help of which it is creating this 5 prime and 5 prime phosphate and 3 prime movement so that this ligation can take place. Okay, so this is directly taken from the lecture slides. So you see here also it has been explained with the help of this figure. So there is a double stranded break, which is causing the recruitment of these two proteins, Q70, Q80, and so the broken ends are recognized. Who is recognizing this broken ends? It is being recognized by this protein 270 or 80, and this in turn recruits the DNA PKCs. Then nucleases. So Artemis is a nucleus which trim the incompatible end. Okay, so for ligation, you need to have two ends which are complementary to each other. So this trimming and is done by the nucleus activity of Artemis. The incompatible ends are trimmed, and then there are several other protein complexes which are recruited one by one. And with the this is XRCC4 DNA ligase 4XLF ligation. So this is a complex protein complex which seals the break, and the double-stranded breaks are repaired. Okay. Okay. Now we will learn about another non-homologous recombination. Okay. So which is conservative site-specific recombination, CSSR. So this conservative site-specific recombination is uh, is a type of recombination between two defined sequence elements. So these are the two sequence elements you can see in this figure. So there are proteins. Now for this CSSR, there are proteins known as recombinases, which recognize the specific sequences where recombination will occur within a DNA molecule. So these are the sequence elements A and B labeled in this figure. So these are the sequence elements, and these are the sites which are recognized by the proteins known as recombinases. Now, this recombinase, what it does, it catalyzes the cleavage and rejoining of the DNA molecule, either to invert a DNA segment or to move a segment to a new site. Okay, so there are several things which can happen by the process of recombination. The segment of DNA that will be moved carries specific short sequence elements called recombination sites where DNA exchange occurs. So the CSSR can generate three different types of DNA rearrangement. One is it can lead to insertion of a segment of DNA into a specific site, as occurs during bacteriophage lambda DNA integration. Second, it can cause deletion of DNA segment, or it can lead to inversion of a DNA segment. So depending on what is the sequence of this sequence element, uh, that will determine whether insertion, deletion, or inversion will take place. Okay. So you see here this figure has been taken from this Benjamin Pierce book, or I think it was taken from Watson, J.D. Watson. So here what you can see is these are the recombination sites. Okay, and these sites are recognized by this enzyme recombinase. So between these two recombination sites, there is a crossover region. There is a DNA sequence which is known as the crossover region. Now what happens? So recombinase will bind the recognition sequences. What are the recognition sequences in case of CSSR? These are the recombination sites. Okay, so these recombination sites are recognized by recombinases. So basically there are four subunits of recombinases. This protein has four subunits. So each of this subunit will bind the to the DNA strand, to the two DNA strands. Okay. And then there will be a cut. The exact mechanism we will see in the subsequent slide. And then you see here that there is exchange of the DNA, exchange of the DNA sequence between the two strands. So you see here that this is one homologous. These are the two homologous chromosomes which have aligned with each other. And here, this chromosome, which is colored in orange, this is getting exchanged with the gray colored chromosome. So this AGC, in place of AGC, you are getting this AGC from the other chromosome. Okay. Similarly, the other DNA strand, through this process, the genetic materials are getting exchanged. Okay. So each recombination site is organized as a pair of recombinase recognition sequences positioned symmetrically. So they're positioned, you can see here, they're positioned symmetrically. Okay. And these recognition sequences flank a central short asymmetric sequence. So this is the asymmetric sequence, and this asymmetric sequence is flanked by the recombination recognition sequences. And this asymmetric region is known as the crossover region, where what will happen, where DNA cleavage. So DNA cleavage will take place at this crossover region, and the rejoining and then subsequently rejoining will take place. Now there are two families of this recombinase protein. Okay, the serine recombinases and the tyrosine recombinases. So why are they called serine recombinases? Because obviously the active site of this enzyme has the protein serine, which is catalyzing the reaction. Okay, and similarly, the active site of tyrosine recombinase will have tyrosine amino acid residue, which will catalyze this recombination process. Therefore, they are known as serine recombinases and tyrosine recombinases. Okay, so you can see here in this figure, look at this figure carefully. So these are the recombinase recognition sites which are present in the two DNA molecules. Okay. So what happens? First, there is a cleavage. A cleavage will take place in the crossover region, which is the asymmetric region. And this asymmetric region is flanked by the recombination sites. So, so you see here a cleavage has taken place, which has generated the 3' OH group and a 5' phosphate group. Okay. Similarly, at the other strand also there is a cleavage. Similarly, in the other DNA molecule, there is double-stranded break taking place in both the DNA strand. Now there is swapping between the DNA partners. So this DNA partner goes with this one, with the upper DNA strand. Okay, and the other DNA strand goes to the bottom DNA molecule. So which results in rejoining and recombination. Okay, so initially the parental strand, if you see DNA molecule, parental DNA molecule had this type of organization, which is marked by this blue colored regions, and after rejoining, creating a different type of DNA molecule, or this DNA, this regions has been swapped, and one of the segments of this DNA strand has recombined with the upper strand. Similarly, the same thing has happened with the lower DNA molecule. So serine recombinase, what it does, it cleaves all four strands. So serine recombinase has cleaved all the four strands, both the strands of this DNA molecule having this crossover region has been cleaved, and similarly in the other DNA molecule, both the strands have been cleaved. So you see here that all the four strands are cleaved by serine recombinases. One molecule of recombinase protein promotes each of these cleavage reaction. As I was mentioning that there are four different subunits of the serine recombinase, and each subunit recognizes one of the DNA strands. Okay. So you have four strands here, 1, 2, 3, 4, and each of these strands are recognized by four different subunits. And yeah, so each of this is cleaved within the crossover region. So where is this cleavage? Cleavage is taking place in the crossover region, and this break is done by one of the subunit of this protein, and these protein subunits are labeled as R1, R2, R3, R4. Now cleavage of the two individual strands of one duplex is staggered by two bases. So you see here, so of the so what is explained over here is the cleavage of the two individual strands of one duplex is staggered by two bases. You see here there are two bases. Similarly, in the bottom strand there are two bases. Okay, this two base region forms a hybrid duplex in the recombinant product. So you see here this strand which is having this two two bases, having this staggered two bases. This will come and join with the this one, this region. And similarly, this one will go and join to this region. And this the other half of the first strand, they will join with each other. So that is why you are getting this type of product. Give me a second. Switch on the charger. So you have to keep in mind that this serine recombinase, what it does, it creates four breaks, four breaks. So four cleavages are taking place in both the DNA molecules, in both the homologous chromosomes. Okay, so there are four strands, and then subsequently strand exchange will occur. Now, tyrosine recombinase is the mechanism of action of this tyrosine recombinase is different. So what it does, instead of creating this four stranded breaks, it is creating breaks in only two strands. Okay, so there are only two breaks that is being produced by the tyrosine recombinase. One is over here and another is over here. Well, in case of serine recombinase, four there are four breaks. Okay. So in all the DNA strands, it has created a break. While tyrosine, it is creating only two breaks. And then similarly, you can see this figure, and this figure, it has been explained very nicely. So after these breaks are created by this tyrosine recombinase, there is exchange. So this part comes and joins with this part. Okay. And then there is a formation of holiday junction in case of the mechanism of recombination by tyrosine recombinase. So you see here that a holiday junction has been formed. Okay, so this part has combined with this part, and this region has formed a connection with this part. So this is called as the holiday junction. Okay, now what will happen? Now this part of this strand, the upper strand, will come in will combine with the upper strand of the lower DNA molecule, and similarly the upper strand of this lower DNA molecule will combine with the upper strand of the this upper DNA molecule. So the bottom strand exchange to finish recombination. So in this way, you are there are recombinants that are getting produced, recombinant DNA molecules that are produced. Okay. So now to answer this question, distinguishes serine recombinase from tyrosine recombinase during strand exchange events. So just now we saw how a serine recombinase differs from the tyrosine recombinase. Now you can answer this question. So as you had seen in the figure also, that serine recombinase was creating breaks in all the four DNA strands. Okay, in all the four DNA strands, there was a cleavage. While tyrosine recombinase was creating breaks in only one of the strands of each DNA molecule. Okay, so that is why the answer is cleavage activity. So here also, in this lecture also, this recombination activity was explained in quite detail. So you can read here. So in mechanism of how serine recombinase causes recombination. So one molecule of the recombinase catalyzes the cleavage of a single strand. So each subunit. So you saw that there are four each of the subunits will recognize each of the subunits will recognize the recombination sites will recognize the recombination sites in each of the four DNA strands. Okay, and once it recognizes and binds to these strands, then what happens? It creates four breaks. Okay. So there are total of four molecules of recombinase which are required for the recombination process. Now, once this break is created, there will be a free hydroxyl 3' end, 3' end, which is that of the sugar. Sugar molecule will have this 3' OH group, and this free 3' OH group is created. And this acts as a nucleophile to attack the recombinase DNA complex at the 5' end to generate free recombinase and recombined DNA. Okay. So this recombinase, so you see here, if you can see this figure, so this recombinase and DNA complex that has been created, that is, it is shown in this green colored circular structure. And so this 5' phosphate has created a complex recombinase, with the recombinase enzyme. And this complex that has been created, this is attacked by the nucleophilic attack of this 3' OH group. This 5' end, the free nucleophile attacks this recombinase complex and gets recombined. Then similarly, what happens? The slippery hydrophobic part of the top and bottom halves of recombinase dimer rotate by 180 degree, and electrostatic interactions stabilize initial and rotated state of recombinase. While in case of tyrosine recombinase, you can see here there are two strands which have been broken, and this recombinase leaves one strand at each side of recombination. Okay. So at each site of recombination, it is creating only a single break. It is only causing a single cut. And this cut strand is exchanged between the DNA molecules. So there's two strands which were cut. The upper strand of the upper DNA molecule and the upper strand of the lower DNA molecule. These are getting exchanged through the formation of holiday junction. So holiday junction was also formed, if you have watched the lectures, this was discussed when homologous recombination was being taught to you. So you see here, so the formation of holiday junction, the two strands are exchanging their genetic material, and then what happens? As the cut at second strand in each side. So first, at one time, only one of the DNA strand is cleaved, and after they have been rejoined, then the second strand is broken, and similarly, this is also recombined with the formation of the holiday junction. And after this, the holiday junction is resolved. Okay, now we will understand the transposition. Okay, I'll cover transposition today. So in transposition, basically, these are jumping genes. I hope you have heard about this term, jumping genes. Jumping genes are transposable elements. So these are there are particular sequences in the DNA or in the genome which can move from one region of the DNA to the other region of the DNA. So you see this figure over here. So this is a particular DNA sequence, and this sequence, this DNA region, which can jump from one side of the DNA to the other side of the DNA. This particular DNA sequence or DNA region are called transposons. These are called transposons, and there are two ways by which these regions can move from one region to the other region. So one is the cut and paste mechanism, and another is the copy and paste mechanism. Okay. So you often do this in your computer. You copy and paste, or you can cut and paste a document. So similarly, Here also the same thing is happening. So this is a cut. So here in this left side, what you see here, this is a cut and paste mechanism, which means that this transposon has has cleaved out from this portion and it has joined to another region of the chromosome. Or what can happen in case of retrotransposons, we will see just now that from this DNA, an mRNA is produced, and then from this mRNA, another cDNA can be produced by the help of enzyme reverse transcriptase, and then this DNA gets integrated to another site of the DNA. Okay, so what is happening? Amplification of this particular region is taking place here. Okay. So this is a copy and paste mechanism. So this the original transposon was at this site, it is remaining at this site, but a new copy has been created at the other side. Okay, but here what is happening? This region has cut out from this part and it has integrated in another site of the genome. Okay, so this is also a form of genetic recombination. Okay, give me a second. Okay, so transposition is a specific form of genetic recombination that moves certain genetic elements from one DNA site to the other DNA site. And these mobile genetic elements, so these are mobile genetic elements because they can move from one side to the other side. And these genetic elements are called transposable elements or transposons. Okay, so movement occurs through recombination between DNA sequences at the very ends of the transposable elements. So in case of CSSR, you are seeing that there were specific recombination sites which were getting recognized by the recombinase, and then this recombinase was creating a break in the direct or in the crossover region, which is present in between the two recombinase site. Okay, and after this cleaving, then the there was exchange of this the DNA regions from two strands, and then ligation was taking place. Okay, but here also there are specific specific regions. So what is this particular regions or what are the characteristic of a transposable element? We will see now. Okay, so the recombination. So movement occurs through recombination between the DNA sequences at the very ends of the transposable element and a sequence in the DNA of the host cell. So recombinants can therefore be identified because the characteristic coded by the inactivated gene is no longer displayed by the host cell. So what will happen if any part of the DNA goes and integrates to another site of the DNA, which is say for example it is coding for a protein, then as we had learned in the earlier lecture that it will cause the that part of the gene to get inactivated. So if it was producing a particular protein which was involved in a function, that protein will no longer be produced. So this recombinance can therefore be identified by this property. Okay, so the transposons can insert within genes, often completely disrupting gene function. They can also insert within the regulatory sequences of a gene where their presence may lead to changes in how that gene is expressed. Okay, so if your transposon if it goes and integrates in a gene which is coding for a protein, then that protein will no longer be encoded and that gene will get inactivated. Or it can happen that this transposon is going and getting integrated in a regulatory portion of the gene. So what do you mean by regulatory portion? It means that the gene can get ligated to or can integrate to the promoter region, or it can go and integrate to the enhancer region. So in this case, what will happen? The expression of the gene will be altered. Okay. So both these changes will alter how the gene is expressed. Okay, so now we will look at the characteristic of a transposable element. So, there are two different classes of transposable element. So one is the cut and paste, copy and paste that I discussed. So one is the DNA transposon. So what are these DNA transposons? So these DNA transposons can be, again, they can be replicative or they can be non-replicative. Okay, so in replicative means what? It is a copy and paste transposition. Okay, so one part of the region is getting copied and then it is integrating to the other region. Okay, a new copy of the transposable element is introduced at a new site, while the old copy remains behind at the original site. So what will happen? If there is replicative transposition, then the number of copies of the transposition element will increase as a result of transposition. However, in non-replicative transposition, it is called cut and paste transposition. The transposable element excises from the old site. Just as we had seen in the earlier figure also, and it will excise from the old site and insert at a new site. So what will happen if it excises from old site and inserts at new site, then there will be no increase in the number of copies. Then there are other transposable elements which can transpose through an RNA intermediate. So that just now I was discussing about that. So these are called the retrotransposons. So these retrotransposons, how do they um, how do they replicate? So from a DNA sequence, an RNA sequence is transcribed from this transposable element, and this is then copied back into DNA by a special enzyme called reverse transcriptase. Okay. So this was your DNA. From the DNA, the RNA molecule is produced, and from RNA, again with the help of reverse transcriptase enzyme, the cDNA is produced. Then this cDNA can go and recombine to another region of the DNA. Now there are transposons. So these are called the class 1 transposons. So remember that DNA transposons can have both replicative and non-replicative, can be of both replicative and non-replicative type, which means that there can be both copy and paste and cut and paste in case of DNA transposons, but retrotransposon will be what? They will always be copy and paste, isn't it? Because this DNA, this transposable element is remaining as it is. It is staying at its original place, while a copy of this DNA is produced and then it is integrating at another site in the DNA. Okay. Now let's look at the structure of transposable element. How will you identify whether in the entire genome this is a transposable element, or this is the region which can jump from one region of the DNA to another region of the DNA? So if you look at the structure of a transposable element, you will see that here there are terminal inverted repeats. So what is the meaning of terminal inverted repeats? So this is, for example, this is your 5' to 3' strand, and this one is your 3' to 5' strand. So this region, the region which is colored in green, if you read it in the direction of 5' to 3', then it will be read as ATC GCA. Similarly, in the other strand, if you read it in the 5' to 3' direction, this is also read as ATC GCA. So similar to what you have learned in about palindromic DNA sequences, that they read the same when read in the same direction. Okay, so this is called terminal inverted repeats. So these terminal inverted repeats will be present in a transposable element, and following this transposable element, there will be flanking direct repeat. So what are flanking direct repeat? So there will be TGCAA. If you see this 5' to 3' strand, then on the upper strand there is TGCAA, and here also there is PGCAA. So if you read in this direction, both are the same DNA sequence. Okay. Here also they are direct repeat. So this sequence is the same two in the two flanking regions. Okay. So there are short flanking direct repeats which are 3 to 12 base pair long. So which are these regions? Short flanking direct repeat, which are colored in pink or magenta, and they are present on both sides of the most of both sides of transposable element. So this is only a transposable element, which means that only this region will cleave and join at any other region of the DNA. Okay, only this region which is marked in green and yellow. Only this has the property to move from one region of the DNA to other region of the DNA. While this region, which is marked in purple, this will stay where it is. Okay, this will not get cleaved or it will not jump from one region to other. So this is a character. This is a signature of the transposable element. So it has direct repeats and it has terminal inverted repeats. Okay. That is why you can see the direction also. So the terminal inverted repeat. So if you read in this direction, in one strand you have to read it in this direction, and on the other strand if you read it in the other direction, then the sequence of nucleotides will be the same. That is why it is called as terminal inverted repeat. And flanking direct repeats means you have to read it in the same direction. So at the ends of many but not all transposable elements are terminal inverted repeats, and these terminal inverted repeats can have sequences ranging from 9 to 40 base pair in length, and they are inverted complements of one another. Okay. If you write the complementary sequence of this upper region, which is ATCGA, so you write the complementary sequence of this region, which is TAGCDT, and then you invert this one. You invert this one, you will get the TCGAT. You will get the other strand. Which is this one, TGCGAT. Okay, so there is ATCGCA. Okay, that is why you get TAGCGT. So if you invert this, you will get TGCGAT, TGCGAT. Okay, so that is why they are called terminal inverted repeats. So if you write the upper strand, the sequence which is present in the upper strand, which is AT, CG, CA. Okay, and then you write the complementary sequence of this strand. Okay, so what will be the complementary sequence of this strand? It will be TA, GC, GT. Okay, and then you invert this. Then you invert this. So this is TAGCGT. You invert it and write it as TGCGAT. So that is the sequence which is present to the other and the other flanking end, which is TGCGAT. Similarly, for this also you can do the same. You can write its complementary sequence and then invert it, and that inverted sequence will be present in the other flanking end. That is why they're called terminal inverted repeats. Okay. So this is the structure of transposable element. Now, one thing might arise in your mind is that how are these direct flanking repeats generated? So so let's understand through this figure. So if you see this figure, then what happens? There are staggered cuts which are made in the target DNA. So what are staggered cuts? Staggered cut means there will be blunt, not blunt end, there will be sticky ends. So if you cut at this position, one of the strand will have this region which will it will have a hover, it will have a 3' overhang, and in the other strand you will find another 3' overhang. Okay, so a staggered cut is produced, and then a transposable element inserts itself into the DNA. So this is that particular site where your transposable element will integrate in this tip region of the DNA. So first a staggered cut is made, and then this transposable element comes and inserts itself into this cut region of the DNA. And once this gets integrated into this region, a gap is left. So who will fill this gap? So this gap will be filled by DNA polymerase. Okay, so utilizing this as the primer, it will extend this 3' this and this having this 3' OH group, and it will extend it. And similarly for the other part also, this gap will be filled by extending this region. Okay, they're extending this sequence, PC sequence. So as a result, you are getting this direct repeats. So replication, you see here a transposable element comes and inserts itself into the DNA, and the staggered cuts leave short single-stranded pieces of DNA. So these are the short single-stranded pieces of DNA which are left. And replication of this single-stranded DNA creates flanking direct repeats. Okay. So remember that these terminal inverted repeats. So, so in the course of transposition, what is happening? The flanking repeats are created when staggered cuts are made in the target DNA. So how are these flanking repeats? How are these direct repeats created? So these direct repeats are created. First of all, in the very first place, what will happen? There are staggered cuts which will be produced, and these staggered cuts will leave short single-stranded pieces of DNA on either side of the transposable element. So this is your transposable element. And on either side of the transposable element, there will be short regions of DNA which will be left free on either side of the transposable element. On the same strand, the two sequences are both inverted and complementary. Okay, so on the same strand. So this is the same strand. So this is one strand. This is the other strand. So you see, look at the same strand, which is the 5' to 3' strand. The two sequences, they are they are both inverted and complementary. While in case of terminal inverted repeats, so the terminal inverted repeats are recognized by the enzyme that catalyze transposition and are required. So this transposase are required for recognition of this terminal inverted repeat. So these transposes recognize these terminal inverted repeat. Okay. Okay, then there are several types of transposable elements. So you will find different types of transposable element in different organisms. So So there are DNA mediated transposition and there are RNA mediated transposition. Now DNA mediated transposition can be both class 1 or they are basically class 2 transposition elements, and they can be both copy and paste and cut and paste mechanism. Follow both these two mechanisms. So in DNA-mediated transposition, you have bacterial replicative transposon, then bacterial cut and paste transposon, and then there are eukaryotic transposon. So there are structural features of these different type of transposon. So in bacterial replicative transposon, there are terminal inverted repeats which flank antibiotic resistance and transposes gene. We will see that the structure of this bacterial replicative transposon. These will have there will be terminal inverted repeats and there are also transposase gene. Okay, so these are autonomous transposable elements because it has a transposase gene of its own. It can automatically come out from a particular DNA region and it can integrate at another DNA region. And the region which is flanked by the region which is between the two direct repeat or the transposable element that will have several antibiotic resistant genes. So the copying of elemental DNA, what is happening in replicative transposon, as we discussed just now, that there is copy and paste, which is having copy and paste mechanism. So there will be copy of element DNA accompanying each round of insertion into a new target site. Particular DNA region will be copied and then pasted into a new target site, which is the example of several examples of these transposable elements have been given on this right hand side of the column, which is TN3, gamma delta, phage mu. Similarly, in bacterial cut and paste transposon, there are terminal inverted repeats that flank antibiotic resistance and transposase genes. So there are antibiotic resistant genes, and these are flanked by terminal inverted repeats. And mechanism of movement is the excision of DNA from old target site. And so cut and paste means it will have the same mechanism that it will excise from one target site and insert into another target site. And there are eukaryotic transposons. Eukaryotic transposons will have inverted repeats that flank coding regions within introns. So the coding regions within the introns. So the eukaryotic genome have exons, have alternating exon and intron sequences. So the coding regions means these exons are flanked by inverted regions, repeats. Okay. Okay. Give me a second.